Hello and welcome to this webinar course on browsing genes and genomes with Ensembl. My name is Astrid and I will be delivering this course together with Emily Perry and Aaron Haskell. We are part of the Ensembl outreach team and we provide courses such as this one online as well as in person. We also look after the help and documentation pages of Ensembl. We run our social media accounts and we run our help desk. So in short, we are the link between our software developers and the people such as you who are using Ensembl. Before I start talking about Ensembl, I need to mention a few technical things. So first and foremost, you have noted that all of your microphones will have been muted. You are very welcome to ask questions during this webinar. If you want to ask questions, please use our Slack workspaces that we have set up for you. My colleagues, Emily and Erin, are both online and will be able to answer questions you have while I'm talking. If you want to reply to a specific person, please do use @username to do so. All materials for this course will be put online under the link you can see here. At the moment, you will find a video from last year on this page, and we will replace this with today's video after this webinar. Likewise, we will also update the exercises and the exercise solutions in the menu on the left-hand side. That way you can find this material for the course online and that there are exercises. There's help with these exercises as well. You can, of course, refer to the solutions you will find on the web page. Also, please use the Slack channels to discuss the exercises with your fellow students and ourselves. And finally, you can also email us at helpdesk@ensembl.org. So now I will start talking about Ensembl, the first part of this webinar. The objective of this whole webinar series on Ensembl is that you get a very good overview about it. So you will hopefully have learned by the end of this course what Ensembl is, what type of data you can explore in Ensembl, how to navigate our browser website, and finally, and importantly, where to find further help and documentation. As I said already, this is the first part of a longer webinar course. I'm doing the introduction right now, and then after a short break today, we will have Emily Perry talking about Ensembl genes. We will then continue on Thursday, the 6th of September, with a variation presented by Aaron, and then comparative genomics presented by myself. And then next week, we will have webinars on regulation, on data export with Biomart, as well as uploading data to Ensembl and the Ensembl REST APIs in the last webinar. And these are more advanced topics already, which go a little bit beyond the browser. Today's session will be structured as follows. I start with this presentation I'm doing right now, and then I will have two demos. In the first part of the demo, I will talk about the homepage and show you how to find information about different assemblies and species. And in the second part, I will demo the region in detail view where you can explore the genome and parts of the genome. Following this demo, we will also have exercises, which, as I said, we will upload to this website for you to do at home. Okay, so the outline of this session is as follows. I will give a brief introduction to genome browsers and then explain the different features and exascales of Ensembl. I will talk about the difference between Ensembl and Ensembl genomes. And finally, I will also talk about the release cycle and give a little bit of background about genome assemblies, because these are important to understand some of the views that we have in the browser. You will all hopefully know that we have been sequencing genomes for a while. The first genome was sequenced in 1977 by Fred Sanger and colleagues, and this was a bacteriophage. And the next big milestone was the finishing of the human genome sequence in 2004. However, between these two milestones, there were, of course, a lot of other genomes finished of different species, and in some cases also of multiple individuals of one species, such as for humans. The question is now, why do we need a genome browser? And my answer to this is this. So a genome is essentially a string of letters, A's, C's, T's, and G's, and we need some way of making sense of this and analyzing it. This is where genome browsers come in. They unlock this code of the letters, and I work from Ensembl, so I will be talking about Ensembl today, but I will not neglect to say that there's also the UCSC genome browser and a map viewer functionality that you can find on the NCBI website. Ensembl is a very powerful resource. It includes genomes and gene builds from more than 100 species, and these gene builds are linked to 3 other types of data, namely variation data, comparative genomics data, so that's alignments, gene trees, and homologs, and also regulation data, and this comes from the ENCODE project. In addition to these types of data, we also have a variety of tools, 2 of which we will cover in this webinar series. The first one is Biomart, that's a tool for batch data export, and the second one is a tool for data processing, which is called the variant effect predictor, and that does exactly what it says and will be covered during the variation webinar. We also have the functionality for you to display your own data, and you can programmatically access our databases via APIs, and Emily will give you a flavor of this in the last webinar of this series. And finally, and importantly, for me really, the best thing about Ensembl is that it's completely open source. That includes the databases which are on the FTP server and the code that is available on GitHub. There are various scales at which you can access Ensembl. If you start by looking at the individual genes or variants, in other words, doing one-by-one searches, you are most likely to use our browser site. However, if you start looking at groups of genes or groups of variants, you will be more interested in our tools, such as Biomart or the web, or maybe the REST API. And in addition, for the people who are really interested in whole genome access, we of course also have a FTP server where you can find all whole genome databases. And finally, there is an underlying Perl API and MySQL databases, which also allow you to access Ensembl at a whole genome scale. And these 2 last methods are certainly the most flexible ones, but they're also the most complicated ones. And this is why we don't cover them in this webinar series, but just so that you are aware that they exist. So in our main Ensembl page, which you can find at ensembl.org, we have more than 100 vertebrate species and also some model organism genomes. If you're very eagle-eyed, you will be able to spot in this tree, we have, for example, yeast, Drosophila, and C. elegans included as model organisms, but also as outgroups for comparative purposes. For the people who are not working on these vertebrate and model organisms, we also have a sister site, which is called ensemblegenomes.org. And here you can find 5 different mini sites, which extend Ensembl across the taxonomic space. So we have a site for bacteria, for fungi, for metazoa, for plants, and for protists. And they all work in a very similar way as Ensembl does, with a minor exception, which I will mention later. Just to summarize that in this table, the main difference between Ensembl and Ensembl Genomes is that in Ensembl, the annotation is done by us, while for Ensembl Genomes, the annotation is done in collaborations with the scientific communities. So we actually only import annotation into Ensembl Genomes. And also Ensembl is 1 year older than Ensembl Genomes. So now a couple of words in our release cycle. I think you will all be aware that data in genomics changes quickly over time, and that's the reason why we operate on a release cycle. So this is roughly every 3 months when we freeze our databases and our interfaces and put out a new release. The one before the current release was put out in April 2018, and that was release 92. At that time, databases were frozen, and then a lot of things happened behind the scenes. What we do is, within these 3 months, we import new genome assemblies, we do software updates, we update our gene sets, we run our comparative genomics pipelines based on the new genes and the new genomes. We also update and add variation data and regulation data. And finally, often we also include new or updated interfaces. And then we have put out a new release, and the current one is from July, and that's release number 93. So this is very important for you. If you work with our own resources, please make a note of the release you are working with, because only then you can go back to this particular data you've started working with. And likewise, if you read an older paper, you can always go back to an older release and have a look at that data from that release. Okay, so now a little bit of background about genome assemblies, because they're important for some of the views in the browser. So a genome assembly is essentially a little bit like a jigsaw puzzle. If you sequence the genome, you generate short reads, and the length depends on the technology you use, but they all have in common that they will not cover a full genome. So what you need to do is you actually need to take these short reads and then match up the overlaps between the reads to get what is called a contig. So this is a contiguous sequence, or just a little, just one string of letters of the genome. Associated with genome assemblies, there's a little bit of terminology you need to be aware of as well. So historically, the human and other reference genomes were sequenced by cloning them into bacterial artificial chromosomes, or bugs. These are shown in these red blocks here. And we call the track of data where we visualize this, these bugs, the tile path track. So a track is a Genome Browser jargon kind of word. It just essentially means a type of data which is plotted along the genome. So now if we take these tile path tracks with the bugs and trim away these overlaps, we find the contigs. We can see the contigs, and these are shown in Ensembl in alternating light and dark blue colors. And then a run of contigs without any gaps is known as a scaffold. And in the final step, we use genetic maps to assemble these scaffolds into whole chromosomes, such as this one. Now, considering this, it's important to note that the human reference genome is actually not a genome of a single person. So in this example here, we have this chromosome on the right-hand side with 4 different contigs, and these 4 different contigs were actually derived from the DNA of 4 different people. So this lady BL's DNA was used to generate this contig BL102 and so on. So yeah, please keep in mind that the reference genome is actually a composite of different pieces of DNA from different people. In Ensembl, we have 3 different human genome assemblies available. The most up-to-date and supported version is called GRCH38. This stands for Genome Reference Consortium Human 38. This is the one you will find in ensembl.org, and it doesn't have any gaps. We also know that in particular in the clinical community, there are people who still work with an assembly called GRCH37. And because there is a community working with it, we have a dedicated website, which you can find at grch37.ensembl.org. This assembly does have gaps, but we support it to a limited amount as well. And we do some data updates. If you read older literature, you may also come across an assembly called NCBI 36, which has 150,000 gaps. We do not support this anymore. We don't update it anymore. However, you can go back to it in our archive sites, which are shown in brown, and we will visit these in a moment. Right. So this is actually the end of my presentation with the theoretical background, and we will have 2 parts of a demo now. As I mentioned, in the first part, we will look at the Ensembl homepage and find information about different genome assemblies and species in Ensembl. And in the second part, we will then look at a specific region and manipulate the view to see any data we are interested in. And just to say right now about the region, you can see I've written this down as 4 colon and then a number minus and another number. So this essentially means we work on chromosome 4, and then we need to use a colon. And this number is the start position, and then we need to use a dash or minus, and then this number is the end position. We need to input our regions in Ensembl in exactly this format. Otherwise, it will not be recognized as a region, and Ensembl will be trying to read it as text, which obviously won't work. Okay, so let's keep that in mind. I will now leave my presentation. I'll actually copy this region. And I will go to my Ensembl Genome Browser for the first part of the demo. And you can find our homepage at either www.ensembl.org, or you can use one of our mirror sites. Today I'm using a US East mirror, and this is why in the address bar of the browser I have typed useast.ensembl.org. Okay, so we are looking at the main homepage of Ensembl now, where at the top you have this blue top header with a logo. If you're anywhere in Ensembl and you click on that logo, it will always take you back to this homepage here. Likewise, this whole top header in blue, in dark blue, will be visible from any page in Ensembl. So you have direct access to our tools, such as BLAST, BEP, other tools, Biomart, also to a download section, a help and documentation section, and a blog. Then on the right-hand side of the homepage, you can see here our current release and when that was released. So this is 93 from July. We have a dedicated tool section up here. And importantly, we have two search boxes. One at the top right, which is visible anywhere in Ensembl, and one here on the front page. On the front page, you have to search for a species first by selecting it from this menu, and then put your search terms into the box. And I say right now that these search boxes in Ensembl are very powerful. They take regions, such as the one I have discussed earlier. They take gene names, they take variant identifiers, they take phenotypes. So it's really worth trying these. So then further down on the homepage, we have a genome section where you'll have some favorite genomes. You also have a drop-down menu here, and you have a link to a full list of Ensembl species. However, the first thing I want to show you from the homepage is how you can get to our archive pages. And for this, I will scroll to the very bottom of the page and follow the link, tiny link here at the bottom right, where it says View an Archive Site. So I click on that, and it brings up a pop-up window with all the different archives we have, which assembly version was used for them, and the dates. I won't go anywhere here, so I will close this window now. In Ensembl, you can close pop-up windows by either clicking on the tick mark at the top right or anywhere outside the window. That does not matter, it has the same effect. So I just click outside this window to close it. And I will now show you a little bit around the species and in particular the human genome assembly. So I will follow the link on the left where it says View Full List of Ensembl Species, which brings me to this page here with a table that you can download using the Excel button at the top right of the table, with all the different species we have in Ensembl, including their common names, their scientific names, their NCBI taxonomy ID, assembly names, assembly accession numbers, and more. So we said we're interested in human, so I will use the filter box at the top right of this table and just type in human there. And then I will click on the common name human, or the picture. And this brings me to the human homepage. Again, now I'm in the human part of Ensembl. We have a search box here now. If I type anything in there, I will only look within the human genome. This line here is important. So this is the genome assembly that we are currently using. And its accession number, just in brackets next to it. You will see that we are working with grch38.p12. So p12 means patch 12, so that's an update to this assembly which was done. It will be incorporated later. And then you have on this page different sections which take you to the different data types I talked about, so gene annotation, variation, comparative genomics, and regulation. But now let's find out a bit more about this genome assembly and annotation. For this, I will click on the link More Information and Statistics that you can find just under the Genome Assembly. And this takes me to this page where on the left-hand side you find some written text about this assembly and how it was annotated, some information about the patches that I mentioned earlier, and also a link to other assemblies. On the right-hand side, you'll have a summary table with statistics and some dates about this assembly, for example, when the gene build was released, how many genes this assembly has, how long it is, and so on. So this is a go-to place for statistics about a particular genome assembly. So now let's say we still work with GRCH37. For this, we could use this link here from the drop-down menu. So we select it here, it's already pre-selected, and click on Go. And this takes me to a page where you will note that the logo now has changed to eBang GRCH37. And apart from that, it looks very similar. So, except that it's in light purple. So this is one way how you can go to GRCH37. There's also a shortcut. If you look at the address bar of the browser, you will note that my US East has been replaced with GRCH37 now. So you can do that as well. And likewise, if you say, I want to go to Ensembl 91, you would replace the GRCH37 with E91. Hit enter, and then you're on this brown archive page. And I won't be saying more about this. So I'll go back to my main Ensembl page, US East, and leave it there for the moment. This is all I wanted to show about Ensembl homepage and species and assemblies, and I will now briefly go and visit Ensembl Genomes. So I will open a new tab, and I will type in the address, ensemblgenomes.org. Which looks like this. Again, we have a news section, in this case with some news about wheat on the left. We have archive sites available for Ensembl Genomes as well, which you can find at the bottom left. And then to the right, we can see the release number and the links to the different mini sites that we have for bacteria, fungi, metazoa, plants, and protists. You can use these links or the ones in the top header to navigate to these pages. So let's say I will go to fungi first, click on that, and I will be taken to a brown fungi page. If I go back to Ensembl Genomes and I click on plants in the top header, I will be taken to a green plants page, and so on. As I said, these work very similarly to Ensembl. So if you are familiar with Ensembl, you will be able to use these as well. There's one exception, which is Ensembl Bacteria, and I will go there right now. And I will now use this little arrow next to the Ensembl Plants logo to directly go to Ensembl Bacteria in the pop-up window. The reason why Ensembl Bacteria works a little bit differently is actually down here. So we do have more than 44,000 genomes of bacteria in this database. And because of that, if you are interested in a particular gene, you will need to search for a genome first and then for your gene. Otherwise, you will be searching the 44,000 genomes and all their genes, and that's just too much. So let's say we are interested in Streptococcus pyogenes and a specific strain of it. For this, I will use the search box to the right and search for this genome by start typing my genome name into the search box. And while I do this, it will start bringing up some hits from our database. I will just continue to type. It's a little bit slow. So now I typed Streptococcus pyogenes. You can see a lot of different strains in assembly, and I will go for this one, AA26216. So I click on that, and this takes me to the home page of this particular strain, which I can now search for a gene using this search box. And if you follow the link to the taxonomy ID here at the top right, you can, by clicking on it, you can actually see the whole classification of this strain in UniProt. Okay, so I will close this, and I will close Ensembl Genomes because that's all I wanted to show in this page. And I will go back to my Ensembl Genome Browser. And we will now start exploring a genomic region in the human genome. So this is the second part of my demo. You remember I said we are looking at a region at chromosome 4 in human. So to go to this region, I will select human from the drop-down menu, and then copy my region into the search box. Just to remind you, we are looking at chromosome 4, and then we use a colon, and then the start position minus end position. And then I click go. And because I put in a region, Ensembl has taken me directly to a view that we call the region in detail view. And this is found on the location tab. So you will notice here at the top left, just under the picture and the human genome assembly, we have a tab now. Ensembl is a tab-based browser. So once you start exploring regions, genes, variants, regulatory features, and so on, you will open more and more tabs. Each tab has a tab-specific menu, which you can find on the left-hand side. So all of these menu items now relate to location-based displays. Before I start talking about this page, I would like to point out one further thing, which is this very important question mark here. So these question marks are found in various places on our homepage, and if you click on them, you will be directly taken to context-specific help for the page you are in at the moment. So I will do this right now, and I opened it in a second tab. And you can see here on this page, there's a description of this region in detail view. I will do this in a moment, but I would also like to point out that within our help pages, we have FAQs here in the menu on the left-hand side. We do have some video tutorials, we have a glossary, and we also have a form to our help desk. So if you click on that, you get to this form where you can put in your name, your email, and your question, and this will go to the Ensembl outreach team, including the 3 people who will be delivering these webinars, and we will answer your questions as soon as we can. I go back to my help page on the region in detail view, and just show you one thing at the bottom. If you scroll to the bottom of the page, you will actually see that on this page we have a YouTube video embedded as well. So here it is. If you click on it, you will see Denise explaining this page.
Hello, I'm Denise from the Ensembl team.
I will stop this because I'm going to do this right now. And I will also close this help page and start explaining this region in detail view to you. So on this page, first and foremost, at the top, you have a chromosome picture with the coordinates on chromosome 4. And the picture has these gray, black, and white cytogenetic bands on the chromosome. There's a red box here. This is the location where we are right now. You can move around within this view by using your mouse to drag a box around a region, and then you get a pop-up window where you can either select mark this region or to jump to the region you would just have dragged out. And I'm not going to do either of this because I want to stay where I am. You may also note that there are these smaller red and on the right hand side also a green highlight on this chromosome. These are alternative sequences that we call assembly exceptions, and they include the patches that I mentioned earlier. So now we have a second picture on this page. In the middle, this is an overview of 1 megabase around the selected region. In this picture, you have an option to either use a drag select tool, which is selected right now, so that's this double arrow, and you also have a tool to select a region by dragging a box. So I can do this in the same way as I've done for the top picture. I will just close this. So here, now we can start looking at tracks. So remember, tracks are types of data that are plotted along the genome. We have a scale bar here at the top, and then our first track represents chromosome bands, shown in gray. And then we have another one with contigs in alternating light blue and dark blue colors. And then we have one with genes, where each of these colored blocks here is a gene, and they have their labels underneath. There's a legend associated with these tracks. So for example, here you have the gene legend. You will see that yellow means merged ensemble or Havana, blue means processed transcripts. And it will become clearer in the next webinar what that means. In this picture, you can also, as I said, you can also use the drag tool. So that's a change to this now. So I use this double arrow, and I can just grab this region and drag it along the genome. And while I was doing this, you will have noticed that the third picture here now grayed out, and I've been given the option to either update the image or to reset the scrollable image. And I'm going to reset it because I don't want to move around, actually. I want to stay where I am. So I click on reset. And I will now go on and talk about this third picture. And this third picture is highly customizable and detailed view of a region. You can move around in the same way as before by dragging or by using the select tool. You have different tracks here shown already. So in the middle, there's a context track with a blue context here. And then we have gene tracks shown above and below these contexts. And just to say right here, everything above this blue line of the context is on the forward strand. So these are forward stranded genes, and everything below is on the reverse strand. So we're looking at reverse stranded genes here. We also have some unstranded features, such as variants, and these are shown at the bottom of the page here. Again, we have legends associated with these tracks, in this case for genes, variants, structural variants, regulatory features. What did I want to say? Yeah, so this is just an overview of what you can show here. You can, if you're interested in a very specific region, you can highlight it. And to do this, use this box select tool that I mentioned earlier, and just drag a box around a feature you're interested in, say this particular exon here, and then you can say mark region. So now I've got a highlight here around this exon, and if I now start dragging this region, I will not lose my highlight, so I can always go back to it. The same applies if I start scrolling. So there's a scroll bar here at the top as well, and also an ability to zoom in and zoom out of this region. So I mentioned earlier this third picture is highly customizable, and to add specific data you're interested in to this picture, you use this all-important configure this page button on the left-hand side. We will use this over and over again during these webinars. So I click on it for the first time now, and this brings up a menu, a pop-up menu with all the different tracks that we can display in this view, and they are categorized, for example, into sequence and assembly, genes and transcripts, and so on. And these numbers here show how much of the tracks we have already switched on and how many there are in total. So we have 3 out of 35 switched on here. So let's say we are interested in proteins and we want to add a protein track. I can navigate through this menu to find it. I personally find it a bit easier to just use the search box at the top left where it says find a track. So I will type into this yellow track, into this yellow box, I will type proteins. And this brings up a couple of options. And I will say I'm interested in proteins from mammals. So I will tick the box next to this track. And this gives me a pop-up window where I can choose what kind of style I want to visualize my track in, and I will go for labels. And let's add a second track. So let's also add some variants, and I will go for some variants from the 1000 Genomes project. So I will type in 1000 genomes, 1000 space genomes, and there are quite a few tracks. The one with the short variants is already switched on, and I will also enable the second one here with the common variants, and I will visualize this in normal view. If you're not sure what the difference between this is, or in fact what any of these tracks show, you can also click on the information button next to the track name. So if I do this here, it will tell me that common variants are variants genotyped by the 1000 Genomes project with a frequency of at least 1%. So I've switched on a couple of additional tracks now, and I will close this window by clicking anywhere outside it or on the tick mark at the top right. And my tracks will load onto my region in detail view now. And that's done. So now we have just at the top above the genes, we have a protein track with proteins shown in yellow. And towards the bottom, we also have an additional track, which is called 1 kg all common SNPs and indels. And this is the one with the 1000 Genomes variants I just switched on. And you can see that there are a lot less of these than for the total 1000 Genomes variants. What you can do within this view is as well, you can rearrange the tracks. For this, you would just grab them on the colored bar on the left-hand side and drag them to a different place where you want to see them. You can also remove a track from your view by clicking on the track name and then the cross mark in the pop-up window. Or you can change the track style by clicking on the name and then the cogwheel and then select one of the other ways of visualization. So I will go for half height here for my proteins, and you will see that the view changes to a more condensed version here. So now you've done a lot of customization here. You may want to actually save this image. You can do this by clicking on this export this image button just above the image. That gives you a variety of options to download this image in different formats. And I'll just close that window. And what you can also do is you can share this page with your collaborator or your colleague next door, or maybe just save a link to it. To get this link with all the customization you have done, you need to use this share this page button on the left-hand side. That creates a unique link to your page with all the customization. That includes, in this case, the mirror here. You can save it or you can email it to somebody. If you use the link from the URL from the address bar at the top of your browser, that will not include the customization you have done. So now a couple of final things on this. You remember this highlight here. So you may want to remove that again to get back to the original view. You can do this by clicking on the pencil icon up here or on the black box above the highlight and just say remove marking. So now my highlight is gone. And finally, you should also, especially if you work a lot with this particular view, should also go from time to time go back to the original display of the page. And you can do this by clicking on configure this page and then scroll to the bottom of the menu and say reset configuration. And also the second one is reset track order. So that now resets my view. You'll see my protein tracks are gone in a minute. So there we go, they are gone. And you can do this resetting also by using the buttons just above the picture here. That's the same. Okay, so that's all I wanted to show about this region in detail view, which allows you to explore genomic region of interest and add some customization to it. I will now go back to my presentation. And I will do a little wrap up before the next webinar. So first and foremost, for the exercises which will follow, just a reminder that you can use the Slack workspace for help with the exercises as well, and we will all reply to you. Erin and Emily, I believe, have responded during my talk already. We will continue to do so and monitor these Slack workspaces over the next days. So here's the link to the course material again. Here on the left-hand side, if you follow this link, you will see that after this webinar, the existing exercises will be replaced by new exercises as well as their solutions. We will also add the recording of today's video and replace the existing video with it. This next page will go to the exercises if you click on that. The Slack channel is not the only way where you can get help with the exercises. You can of course use these solutions as well. And as pointed out earlier, please feel free to email us at helpdesk@ensemble.org. As I mentioned earlier, this is the first in a series of webinars on the Ensembl Genome Browser. After I finish this one, we will have a 5-minute break, and then Emily will continue with Ensembl Genes. And then on Thursday, there will be Erin and myself talking about variation and comparative genomics. Next week, we will have more webinars on regulation, on data export with Biomart, and finally on the 13th of September on uploading data with Ensembl and the REST APIs. So as I just said, the next webinar will be delivered by Emily. She will be talking about the Ensembl gene annotation system. We provide annotation for selection of vertebrate genomes and also for some non-vertebrate model organisms. In this webinar, you will be able to learn about the methods for gene annotation that we use and also how you can assess the quality of different transcripts. Emily will introduce the system of Ensembl stable identifiers, as well as a system called gene ontology that allows you to describe and categorize protein function. And this webinar will start after about a 5-minute break, just after this one. Just a few more things, more general things. There's a lot of online resources available about Ensembl where you can get further help in documentation. I mentioned the EBI training website where you can find this webinar course already. We also have a dedicated Ensembl tutorial page where you can find Ensembl-specific tutorials. We have a YouTube channel where you will be able to find these webinars as well. And this also includes taught videos on shorter topics. If you ever find yourself in China and have problems accessing YouTube, you can access these on Youku as well. I mentioned our help desk already, where you can contact us with questions or suggestions at any time. We also have 2 public mailing lists. One is the developers mailing list, or DEV, where developers outside and inside Ensembl discuss their more technical problems, and we have an announced mailing list where we talk about new releases and what we plan for new releases, new software updates, new features, and so on. We have social media accounts where you can follow us. So we have Facebook, we have a very active Twitter account, And we also have a blog that you can find at ensemble.info, where we write about new releases, where we introduce team members, where we talk about new features or just general news. Ensemble is a completely free resource, so it's very important for us that you cite us if you use it. You can find our complete list of publications here with the main paper from 2018, and there will be various papers mentioned throughout the workshop on more specific topics as well. This is the whole Ensembl team during our retreat earlier this year. We are at any time between 60 and 70 people. Most of them are software developers and database people, and the outreach team just has the pleasure of going out and talk about all the hard work that these other people do. So finally and importantly, I need to acknowledge the whole Ensembl team, which is listed here, and of course also our funders. Our main funding comes from the Wellcome Trust, but there is a variety of other funding resources, including Ensembl, that also support us. So yeah, thank you for your attention. That's the end of the webinar, and we will be having a break of about 5 minutes now, and then Emily will carry on with the next webinar on Ensembl genes.